You've got a very large variety of different types of cells in your body, but they all sort of have similar, well, same basic functions at the same time, all carried out by key macromolecules. So some example of functions are to synthesize material, release energy, used in storage, used to transport other molecules, and used to store or transport instructions to other cells or to the cells itself, or the machinery within cells. And here are our 4 key macromolecules. So you've got your carbohydrates with the example of glucose or amylose. You've got amino acid, which is like alanine and cytosine, which join together to form proteins. You've got nucleotides, which can be things like adenine and ATP. These join together to form things like DNA. You've got your lipids, which are things like triglycerides and cholesterol, but these aren't traditional polymers, so because they're not made up of repeating units, so they're not polymers. All your carbohydrates have the general formula of Cn and then H2O in brackets with a little n. This is basically, you have the same amount of carbons and oxygens in a carbohydrate, but then you have double the amount of hydrogen. So these are important in releasing energy through things like glycolysis, storing this energy in the form of amylose and starch. It can also be used as structural polymers such as your cellulose in plant cell walls, and also cell-to-cell interactions by forming receptors on the cell membrane. So you've got your glycolipid receptors. They have 3 layers, the simplest being the monosaccharides, which are just one molecule. This can be your glucose, your fructose or your galactose. You then have disaccharides, which consist of 2 monosaccharides joined together by a glycosidic bond. So for your sucrose, this is made up of glucose and fructose. Your lactose is made up of glucose and galactose. And your maltose is made up of 2 glucose molecules. You then have polysaccharides, which are long chains of monosaccharides, some examples being amylose, glycogen and cellulose. But how do these glycosidic bonds form to form disaccharides and polysaccharides? Well, it's through something called condensation or dehydration. It's when a hydrogen group and a hydroxyl group of the 2 monosaccharides sort of join together and are released, forming a water molecule, so a H2O molecule. Where these missing groups were, the glycosidic bond forms. So because you don't have the hydrogen or the HO or OH anymore, a bond can form between the 2 there. That's why it's called dehydration, because you'll lose a water molecule. If you rehydrate the molecule and add water again, hydrolysis occurs and the glycosidic bond is broken, forming 2 new monomers, and the polymer is broken. So onto our nice little nucleotides. These can act as a form of chemical energy and to store information. So it's often made up of a phosphate group attached to the 5th carbon of a ribose ring. You then have a nitrogenous base attached on the other side in the form of a purine or a pyrimidine. Your purine is formed when a ribose ring is phosphorylated. A purine is then added, and then as is a phosphate. In the pyrimidine pathway, it forms a pyrimidine group, adds a ribose ring, and then adds a phosphate. So if you have 2 adjacent nucleotide monomers, these condense together to form a phosphodiester bond between the phosphate group and the 3' carbon of the ribose ring of the other nucleotide. As this is condensation, again, a hydrogen molecule is lost. If this occurs between multiple different nucleotide monomers, you form a polynucleotide, such as your DNA or your RNA. So you don't really need to know, but there are 22 proteinogenic amino acids, even though we say there are 20. And there are some which are coded for just by prokaryotes and some which are coded for just by eukaryotes. It can result in multiple different types of responses and functions, such as enzymes, adaptive immunity, in messaging and in structure. Your amino acids are condensed together to form a peptide bond, which makes them into a long chain of amino acids. This is called the primary structure of a protein. It's the number and sequence of amino acids. They can then undergo folding into beta-pleated sheets or curling into alpha helices. This is a secondary structure. These can then fold into a specific 3D structure. It's due to things like the charges of the air groups causing interactions. This is the tertiary structure. If multiple polypeptide chains of a tertiary structure sort of join together, you can get a quaternary structure protein, such as hemoglobin, which is made of 4 different tertiary structure chains with a heme or iron group in the middle. Last but not least, we have lipids. These are long hydrocarbon chains with a carboxyl group attached to the end of them, or the beginning of them really. These hydrocarbon chains can either be saturated, meaning there's only a single bond between each carbon, or they can be unsaturated, meaning there may be a double bond between the carbon. It's because of this double bond we say that they can either be cis, so it means that because the carbon can then only have 1 hydrogen attached to it instead of 2, they will both be on the same side, so they will both either be like on the bottom or on the top. Or they can be trans, where a hydrogen will be, one will be on the top and one will be on the bottom. I'm not really going to go into that much detail about this though. One thing I am going to mention about these hydrocarbon chain tails is that they're hydrophobic. That means water hating. So because of the many non-polar covalent bonds within them, they actually repel water and are non-soluble in water, but they are soluble in organic solvents. 2 types of lipids I'm going to talk about are your triglycerides and your phospholipids. Triglycerides are made up of a glycerol joined to 3 hydrocarbon chains by an ester bond. These chains are electron-rich and anhydrous. Your phospholipid is very similar, apart from it only has 2 hydrocarbon chains and there is a phosphate head. That means that it has a hydrophilic, which means water-loving head, and a hydrophobic or water-hating tail. Quick note to add, the fact that these tails are electron-rich and anhydrous, it means a lot of energy is actually released when they're oxidized. But if we go back to phospholipids for a second, these are very important in making plasma membranes because they can form a phospholipid bilayer. So the hydrophobic tails will face inwards away from any water or fluids, whilst the hydrophilic heads will face outwards into the fluids, forming sort of like a barrier to separate them. Another type of lipid that I will mention are steroids. These aren't actually from fatty acids and they're not hydrophobic, but they're still classed as a lipid. And we've addressed each of these macromolecules sort of as individuals, but they can actually conjugate with each other. So for example, you have oligosaccharides, which can conjugate with other molecules. We sort of have glycolipids, which are a lipid and a carbohydrate. You have glycoproteins, which is a protein and a carbohydrate. And there's also recently been a study on glycoRNA. So it's a conjugated molecule of RNA. I've actually just put the title of the paper there for if you want to go and have a look. It's really cool.